Hello and welcome to today's webinar organised by the Ophthalmologist and ALPAL. My name is Mark Hillen, I'm the editor of the Ophthalmologist, and it's my great pleasure to be the moderator of today's online event, which is entitled Adaptive Optics in Ophthalmology. So, please let me introduce today's speakers, Dr. Ramkumar Sabesan, Dr. Robert J. Zawadzki, and Dr. Jennifer Hunter. All speakers will be on hand to answer any questions that you may have at the end of the presentation, but please remember that you can submit questions at any time throughout the webinar. Our first speaker today is Dr. Ram Sabesan at the University of Washington. Ram was born and raised in New Delhi, India. He earned his bachelor's degree in engineering physics from the Indian Institute of Technology in Delhi. He earned his PhD at the Institute of Optics at the University of Rochester, developing non-invasive optical tools for the study and treatment of corneal pathologies. Following his doctorate, he did a postdoctoral fellowship at the University of California, Berkeley, studying the retinal basis of color perception using advanced high-resolution imaging. Our second speaker is Dr. Robert J. Zawadzki of the UC Davis Eye Center. A graduate of the Nicholas Copernicus University in Toruń, Poland, he left with a BSc in experimental physics and computer sciences and an MSc in medical physics. Robert received his PhD at the Technical University of Vienna, Austria. In 2004, he began working with John Werner, and he later co-founded the Vision Science and Advanced Retinal Imaging Laboratory at the Department of Ophthalmology and Vision Science, UC Davis, Sacramento, California, where he is right now. In 2012, together with Professor Ian Pugh Jr., he co-founded the UC Davis RISE iPod, a small animal retinal imaging laboratory, and he became its managing director. His research interests focus on studying microscopic, i.e. cellular resolution morphology and function of human and small animal eyes, as well as the dynamic changes in various types of retinal and optic nerve head diseases by using high-resolution in vivo imaging modalities AO-OCT and AO-OSLO, including its combinations and functional extensions. Our third speaker today is Dr. Jennifer J. Hunter of the University of Rochester, where she is an assistant professor of ophthalmology and she holds secondary appointments with the Center for Visual Science and Biomedical Engineering. She received her PhD in physics and vision science from the University of Waterloo in Ontario, Canada, as her current research focuses on the mechanisms of light-induced retinal damage and non-invasive fluorescent imaging techniques to study retinal function in healthy and diseased eyes using AO-OSLO. She is a member of several committees of the American National Standards Institute, the Standard for the Safe Use of Lasers, and is the chair of the Bioeffects Technical Subcommittee. So, let me get today's webinar underway by passing you over to Ramkumar. Ram, over to you.
Thanks, Mark, for the introduction and for the invitation to speak here today. Our lab's focus is to design and use adaptive optic systems for the study of human visual function. Briefly, we develop novel optical systems and use these systems to ask how our most fundamental features of vision arise and how these facets of vision suffer in the face of retinal diseases. Okay, so we and a host of other labs are interested in the fundamental aspect of neuroscience and in how to link visual behavior, color, motion, etc., to the activity of isolated neurons in the visual system. Here, the classical Nobel Prize-winning Hubel and Wiesel paradigm is still very prevalent, where an animal or a human is visualizing a stimulus, moving bars, colored gratings, and so on, and the experimenter uses methods such as electrophysiology, calcium imaging, fMRI, or psychophysics, or a combination of a few of these to relate which cells or biological features of the visual system is firing, lighting up, etc., in response to the stimuli. Ideally, we'd like to translate all or a large part of these methods to the human and identify the cellular substrates and computations that give rise to fine visual perception. Furthermore, if we can understand the normal visual system, we can dream a little further to even translate a few of these to the clinic to evaluate whether we can find the mechanisms of diseases and treatment outcomes in patients. So let's look at a cartoon very similar to the picture of Hubel and Wiesel to appreciate what might be desired to translate classical physiological techniques to the human. Let me just say at the outset that we are indeed very close to achieving some of these things in practice. For the sake of simplicity, I'd like to describe our quest here as three parts. Seeing cells, as in we would like to visualize the structure, morphology, the function of the neural cells we wish to study. For this, we desire spatial resolution, non-invasive imaging, high signal-noise ratio, etc. we'd like to be able to stimulate the cells and perturb a cell and see how it reacts to this perturbation. A simple example of this would be, say, for instance, how cones react to light stimuli. Here we desire selectivity, specificity, and repeatability, know which cell is hitting, and to be able to do it again and again. Finally, we'd be able to record response of the visual system, either at some intermediate points in the visual system, say in the retina, the LGN, the cortex, or study an aspect of visual behavior, such as light sensitivity of individual cones in the retina, or the color percepts they provide. This is an overtly simplified picture, and of course, there's more to this, but for the purpose of this talk today, we can think of this as our roadmap. And first, we can just start with how we can begin to see cells in the retina, where adaptive optics turns out to be the most transformative tool. The retina is situated inside the eyeball, behind the cornea and the lens of the eye. In order to visualize the retina in vivo in an intact eye, we need to peer through these optical components, the cornea and the lens. How good or bad these optics are and what optical constraints it poses on imaging features inside the eye is where AO comes in handy. So this slide by Austin Rohde from UC Berkeley summarizes well the need for adaptive optics for the eye. The point spread function, or the smallest point achievable at the image plane, scales inversely with the size of the entrance pupil, and this is true for the eye as well. The numerical aperture of the eye for a 7 millimeter dilated pupil is sufficient to resolve features on the order of about 2 microns inside the eye. The caveat, however, is that this is true only in the case of a system whose performance is limited just by diffraction alone. In the case of a typical eye, however, the optical performance is limited by the imperfections in its focusing elements and not by diffraction. This leads to larger, blurry point spread functions as the pupil size increases, as you see in the southeast part of the slide. In order to elicit the maximum benefit achievable given the anatomical numerical aperture of the eye, we need to adopt techniques which correct for these imperfections, and the suite of these techniques all fall under the umbrella of adaptive optics. So the seminal paper that first demonstrated adaptive optics for the eye came from the University of Rochester, when Junjong Liang, Don Miller, and David Williams applied it to achieve 2 remarkable feats. First, they demonstrated images of cone photoreceptors in a living eye. And second, they demonstrated that once the eye's optics are corrected, individuals achieved significantly improved vision, allowing them to see extremely high spatial frequency features, which were otherwise filtered away by the eye's aberration. This current year, 2017, indeed marks the 20th anniversary of this particular publication. So our lab's overarching efforts are exactly to link these 2 applications of AO and ask how individual cone photoreceptors and their ensuing circuits mediate our most fundamental senses of vision. And we'll talk about that just in a second. Since its inception into fundus cameras and vision correction setups by the Williams lab in the 90s, variants of adaptive optics have been applied to different imaging techniques and to improve image quality across all these modalities. But today, I'll focus only on vision simulation correction and the confocal scanning laser ophthalmoscope. It is, however, important to note at this point that adaptive optics is not an imaging modality in isolation, in that it's just a tool that can be coupled with standard imaging modalities such as OCT, fundus cameras, SLOs, etc., to allow for microscopic, diffraction-limited access inside the eye. So how does it all work? Well, the distortions in the cornea and the lens of the eye is sensed with a wavefront sensor, typically a Schock-Hartmann wavefront sensor. The sensor then sends signals into a wavefront corrector, in this case, a deformable emitter, which can induce equal and opposite distortions as to those present in the eye to achieve diffraction-limited resolution. Once this optical system, of which the eyeball is now an integrated part, is well corrected, it can be used to either image structures inside the eye or use fundus imagers, SLOs, or OCTs. On the other hand, an external CRT monitor projection system, your latest AR, VR toy, can also be used to shine sharp, well-corrected features inside the eye. So at this point, I'll spend just a few seconds talking about the latter aspect of this technology, the visual function testing, by illustrating how this cartoon, the schematic, might be implemented in practice. Note here that from the eyeball, pairs of lenses, relay lenses, are used to achieve optical conjugation of the eye's pupil, the deformable mirror, and the wavefront sensor. A wavefront sensing beacon is used as a light source to measure the imperfections. And note that there are no detectors in such a device, and that the retina of the human person itself is the detector. The Hubel and Wiesel television monitor or projector may be used for displaying stimuli. Extra pupil planes or Badal systems may be used to compensate partially for sphere and cylinder. Note, while this is just an example, the core of a system used to measure visual function with adaptive optics without any imaging follows very similar principles. So as you can appreciate in this plot on the right, the number of studies using adaptive optics for measuring aspects of visual function has seen an exponential rise since its inception in the 90s. While this plot is now 7 years old already, adaptive optics as a technology still remains nascent if you consider how long techniques such as confocal microscopy has been used in biology. Using these tools for measuring visual function, a number of different questions have been asked and addressed by us and others. So some of these questions are, what are the limits of vision? What are the benefits of vision when we correct aberrations? They're being used to test drive custom vision treatments for eyes with really high amounts of aberrations. They're being used to devise new ways to improve depth of focus in presbyopic patients. And finally, the one that's most interesting to us at this time is to study the interaction of optical and neural factors that affect vision. While measuring visual function without imaging certainly has its advantages, correlating them precisely with measures of retinal structure, such as those derived from OCTs or for fundus imaging, stand to inform us most about the underlying physiological processes in both normal and diseased retinas. So visual acuity, contrast sensitivity, microperimetry are some of the most clinically relevant tests of visual function and are being used routinely. Adaptive optics here offers two advantages for applying these to understand disease mechanisms and treatments on a cellular scale. First, we can use adaptive optics to make the size of the stimuli as small as the size of the cell you're probing, for instance, the size of a foveal cone. Secondly, simultaneously, you can image the same patch of the retina that you're stimulating with the stimulus, such that you can draw direct correlations between the structure of the cone or the structure of the cell that you're probing and the function that you're testing psychophysically. Let me elaborate this a little further. So visual stimuli can be delivered through adaptive optics correction in conjunction with imaging by precisely modulating a laser in a given pattern, in this case, an E. In this manner, a record of the stimuli can directly and unambiguously be embedded into the video stream and inform us how the stimuli has interacted with exact cells and their temporal characteristics. However, in a lot of cases, we'd like to learn how the stimuli has interacted with just one cell in particular. And for that, eye tracking algorithms have been developed which allow the tracking of features within the retina which are just as small as the scale of individual cones. Evidently, this cone stuck between the crosshairs in this movie demonstrates how precise this eye tracking is. Now, finally, combining the first and the second feature here gives one the ability to track and target the same cells in the retina repeatedly of human observers. The best way to appreciate this feature is if you notice how the cones stuck between the forks of the E remain confined to the same features as the eye is continuously moving. This now has given us the opportunity to probe characteristics of human vision with stimuli that can be tracked and targeted onto individual retinal cells. While I do not have the time to get into the details here, I do not want to undermine that this capability of retinal tracking and targeting with high spatiotemporal precision is a result of a decade of engineering performed by Austin Norda of UC Berkeley and his collaborators. Okay. So now we wish to use this technology to address one of the most fundamental functions of the visual system, one in which the retina is thought to play a really major role, that of color vision. Color vision poses an interesting puzzle for vision in that on a local scale, the cone mosaic, the trichromatic cone mosaic is colorblind. Take this particular M cone shown with an arrow for an example. The two wavelengths at about 500 or 600 nanometers elicit the same excitation in this M cone, leaving the brain to figure out tricks to reduce the color associated with them. Similarly, a small amount of 550 nanometer light wavelength produces the same excitation as a large amount of longer or shorter wavelengths, leaving a confound between luminance and intensity, luminance or intensity of the light, and the wavelength of the light that's shining on the retina. So in order to address these puzzles, we would first like to be able to delineate the type of cone, long, middle, or short wavelength in a human, then target them in ways that excite specific retinal pathways or circuits, and finally ask the subject what color he sees when that particular cone is targeted. So one of the first scientific applications of adaptive optics when it first became available for human imaging was to map the cone types in a human trichromatic cone mosaic. And this was done by combining traditional classical retinal densitometry, but coupled with an adaptive optics fundus camera. Since then, these fundus cameras have given way to scanning laser ophthalmoscopes. And more recently, we have used an adaptive optics scanning laser ophthalmoscope to achieve similar cone mosaics in which the three cone types are labeled. Then we can go back to the same mosaic and record the color percepts when individual cones were stimulated. Here, 543 nanometer flashes were shown 20 times each to a cone, and the subject was asked to categorize his perception of colors into one of these 6 categories, either green, white, blue, yellow, red, or not seen. Here, it's important to note that a 543 nanometer flash looks green in hue or color, while, as I've mentioned before, the cones are indeed blind to the kinds of wavelengths that are shining on them, as we'll see in the next slide. Because we know with a good degree of certainty where the stimulus landed on the retina and what color was associated with it, we can associate the delivery of every light flash with a spatial location and color percept. For these 3 example cones, we see how one of them is dominated by a green response while the other 2 are dominated by reds, with a little bit of white or achromatic response in each case. We can convert these bar charts into what we call donut histograms, representing the distribution of color in each of these cases. Now we combine the 2 pieces of information we've had so far by labeling the cone types, long, middle, and short wavelengths, with their own individual donut histograms. Shown here are data from 2 subjects and roughly about 250 cones. This all in itself is an extremely rich data set and allows us to draw direct correlation between the activity and spectral type of the cone and its associated percept. From here, we can go ahead and ask a few fundamental questions. The answers, and you can read more about them in detail in our paper. For instance, we can ask, what is the consistency or the purity with which a cone signals a percept? When it does signal a percept, is it always a colored one? In general, we find that pathways underlying these cones are remarkably consistent in their signaling percepts, and that only a small handful, about 1 in 3, signal hues, while the rest of them signal white or achromatic percepts. You can further begin to draw relations between spectral neighborhoods of a cone and so on, all of which let us have deeper insight into how the retina mediates our most fundamental aspects of vision. So to learn more about the study, you can go ahead and read our paper, which came out last year. So with that, I hope I have given you a flavor of how adaptive optics is a key ingredient in giving us unparalleled access to the visual system and letting us now do the kinds of things or experiments in living humans that physiologists have been conducting in tissue preparations for many decades. The future for us certainly involves applying such techniques to probe disease mechanisms and etiology on a cellular scale and establishing more sensitive biomarkers. This work, the second half of it, was all done in the lab of Boston Rhoda in very close collaboration with Brian Schmidt, a postdoc in the lab. I'd also like to thank our other collaborators for input both at Berkeley at the University of Washington and also for our funding sources. With that, I'd like to thank you for your attention. If you have more questions, please email us. And with that, I will pass it on to Robert Zawadzki.
Thank you very much. So I'm also I want to thank for this invitation opportunity to present my work here, and I'll just start in a second. Okay, so today I will talk about an AO-OCT in vivo cellular resolution imaging in the retina, mostly about clinical applications and future directions. The talk is divided into three kind of major portions. One is a very brief introduction to AO-OCT. Then I will also try to briefly go over clinical applications of this technique. And at the end, hopefully time allows to give you some details about the very exciting future directions of this technique. So next about AO-OCT. So first about OCT itself. So the OCT stands for Optical Coherence Tomography, probably very well known in the ophthalmic field right now. It basically allows you to measure cross-sectional images of the retina or an object. In ophthalmology, it's mostly anterior chamber or the retina. And here what you can see is that we're measuring depth scattering profile as a function of depth here. That's so-called A scan. And then this can be used to reconstruct a B scan, which is created from the series of this A scans. That would be the OCT B scan of anterior chamber. And for an OCT system, the lateral and axial resolutions are decoupled. So axial resolution depends on the properties of the light source used, and the lateral resolution would depend on the optics that is used for imaging. And in case of the eye, as you may imagine, this would also depend on pupil size and application of adaptive optics. And then if one wants to scan the back of the eye, retina, then we have to basically go, we'll jump through. We basically have to scan in the radial pattern. So with the OCT being now around for over 20 to 25 years, you can get really powerful clinical tool that allows you to measure the cross-sectional view of the retina, as on this slide. That basically shows you on the left is a schematic of OCT. On the right, you can see the cross-sectional view with the labeling of different retinal layers, and this can be also used to reconstruct the volumetric information of the retina, including segmentations and thickness maps, and that's more or less standard of care for the OCT these days. The difference between the AO-OCT and OCT is that we basically have to introduce an adaptive optic control system inside our OCT. So what you can see on the slide here is that the small inset on the right top corner shows you an OCT system, and the bigger one is basically how OCT has to be changed in order to allow AO-OCT. So we basically have to include this AO control loop with the fiber mirror, wavefront sensor, and some kind of reconstructor to basically allow us to correct aberrations. So this is the part that is common for all AO systems. So there's no difference in AO-OCT. And if everything is working, we can achieve resolutions in the order of 2 to 3 micrometers in all 3 dimensions. So in this respect, AO-OCT is unique, that you're really getting a really high resolution volumetric data set. So this slide shows you how the turning on AO looks like on AO-OCT B scans. So as you can see, basically when we're turning on AO, depending when we set the focus, we can either focus on photoreceptors. That's the case on the left image, or we can focus on the inner retina. That will be the case on the right image. So as you can see, the OCT really gives you the cross-sectional view, and AO gives you this really high lateral resolution. So if you go to the next slide, this is basically a comparison of the different point spread function offered by, or resolution offered by different imaging systems. So this is a slide borrowed from Don Miller, but what you see here is that the different patterns on the right shows you the kind of the minimum size of the sample that you can acquire from the object, and basically in the case of AOCT, we're getting the smallest volumetric point spread function. So this basically corresponds to the best kind of three-dimensional resolution you can get. And then if you go to the next slide, this is a comparison between the kind of imaging feature offered by AO-OCT and AO-OCT. So AO-OCT is this classical view of photoreceptor mosaic, where one basically acquires images in a C-scan mode when you can see individual photoreceptors very well. In case of AO-OCT, the major acquisition mode is in this B-scan. So basically, we're getting this cross-sectional view of the retina. So in this case, this is basically visualization of the photoreceptors as seen by AO-OCT on the left, and then as seen by AO-OCT on the right. So instead of seeing this nice mosaic on and fast view as seen on AO-OCT, AO-OCT really gives you this cross-sectional view of photoreceptors. So we basically start seeing individual reflections from within single photoreceptors, and we basically right now assigning this reflections to the inner segment outer segment junction of a single cone, and then the end tips of the outer segment. So so one of the development highlights over years for AOCT was to combine this with other modalities. This is just one example where we try to combine AOCT with AOSLO, and just to show you that in a way these two modalities are really complementary. You can see here that the example of the simultaneously acquired AOSLO image and AOCT image, and then the next slide basically shows you the live video of the system when the AO is turning on and off, and you can see increased brightness of the photoreceptor mosaic as seen on AOSLO, and then increased visibility of the photoreceptors in the outer segments and outer segment tips with the AOCT. So here, what I also showing you is that the first image is on the left is AOSLO, the middle one is AOCT in a linear scale, which corresponds to the AOSLO image. Then on the right, you have a logarithmic scale AOCT image. The next thing I want to kind of point out is that the early days AOCT systems were probably not as careful design in a way that we actually suffered from some system aberrations and some people wandering. And what we did in our lab, and also some of our collaborators, Don Miller, we basically following up the steps of AOSLO community, we started developing and working on the aberration-free system. So this is just an example of the change in the optical design of a kind of old system and the new type of system. So with these systems in hand, you're basically getting much better optical performance, and we really can focus all the efforts of adaptive optic systems on correcting just aberration of the eye. And this indeed results with a much better performance of the system. So we actually we start resolving in these volumetric acquisitions the structures coming from cones and rods and other structures. And this is something that it's still a kind of ongoing work in the AOCT community. This is just an example of how the image of the photoreceptors, rods and cones looks like on an AOSLO image, and this compared now to the image with the color-coded cones and rods from AOCT image. So next few slides, I'll basically show you some of the clinical applications of this technology. So one of the applications is clearly use of AOCT for just imaging structures. In this case, there's a slide showing you the image of the optic nerve head acquired with the AOCT. So we really start seeing a kind of detailed view And again, the AOCT is intrinsically volumetric technique, so we can acquire volumes. In this case, there are just single volumes without any averaging. Another way to look at to use AOCT in clinic is basically try to have a kind of high quality AOCT B scans that really allows us to follow up the structural changes at the cellular resolution. So this is an example of evaluation of the healthy subject. And what you can see on top, you're basically seeing data acquired from the subject with different modalities, fundus camera, multifocal ERG, microperimetry, and in this case everything is perfect. This is a healthy subject with no vision loss, and you can then look at the green lines on the panel D being locations of the AOCT scans shown at the bottom of the slide, and then the same paradigm was applied to image patients, and in this case you can start seeing a kind of really detailed view of the layout of different layers in the retina, being photoreceptors or RPE. And in this case, basically, we're getting this much more detailed view of the structural changes in the retina, correlated with the functional measured by different techniques that are used in clinics. And the next slide is another example of another patient with late-stage AMD that you can clearly see huge disruptions in receptor structure and also function of the retina. Another interesting application of AOCT is basically this is the work of my colleague Ravi Janal. Is basically trying to understand what is the cellular structure, what are the cellular structures responsible for creating of the image seen by OCT. And there's specifically some controversy about assignment of the layers of the photoreceptors in the retina, in retinal OCT. And Ravi basically made a very nice work shown here, published in IOVS, where he basically showed that the reason why the photoreceptor layers look so thick on clinical OCT compared to the AOCT is basically that the systems are producing highly blurred and average images. And what really is a very kind of sharp reflex from all of these layers appeared very blurry, and this is probably a reason why there was some confusion in the field. But I think right now this is again an example of a zoom-in view of the OCT and AOCT, and so right now we are basically pretty sure that the assignment of the layers should be as follows. The first thin layer should be the ELM, then we think should be actually inner segment, outer segment junction, then the COST stands for the cone outer segment tips, and then the last layer is the retinal pigment epithelium. And Ravi basically was able to acquire multiple volumetric AOCT data sets when he analyzed the length and thickness of each of the reflectors of the layers seen on AOCT, and then compared it with an average values from OCT, and clearly showed that they have the same positions, but they're much thinner, probably because we don't average this data in AOCT systems. And that's, again, analysis of multiple thickness of these bands, and based on that, we basically think that at least the band number 2 is really a result of reflection at the inner segment, outer segment junction when you have a change in reflective index inside photoreceptors. That would be the band 2 assignment. So I want to finish with Ravi 4 very exciting new directions that are really fresh, and some of these papers were just published this or even or last year. So the first, and they all kind of follow the direction of using AOCT as a functional tool to measure the retinal function. So the first is really amazing job done by Don Miller's lab and published by Omer Kokaoglu, where Don basically was able to acquire multiple volumes of the retina over and over again over minutes, and he observed that if you choose from each volume one specific photoreceptor, in this case cone, and if you try to look at the change of this cone reflectivity over time, what he was able to do, he was able to observe that the location of this third band that I showed you before. So the ISOS is this inner segment outer segment junction. But if you look at the right bottom side of this image, there is this band that we assign to the cone outer segment tips. So if you look at the temporal change of this band, so again, we're looking now at the single cone over time, you can clearly see that there's disruptions, and for some of these cones, the cone outer segment disappears, even though the inner segment outer segment junction is completely static. And I think don't properly assign this effect to the photoreceptor disk shading process. And now with this tool in hand, he basically can analyze changes in photoreceptors disk shading in vivo in humans. And this is an example of a very, very beautiful analysis showing you first on the left-hand side, there's all the cones that were found within the volume that were then followed over multiple hours, over 1 day time. And then each of this cone then is labeled based on when the shading was observed. So you can see there's increased number of shading in the morning, that's yellow, and then much lower number of shading in later time of the day. You can also follow up for different subjects and look what was the change in length, and this is the histogram showing you that. So basically, this technique allows detailed study of the shading process in living human eye. The next really interesting application for AOCT is mainly combining AO with optical coherence tomography and geography, and this is also recent work by Michael Pierscher. So in this case, OCT and geography is also one of the very important development in clinical OCT, where one can get access to the vasculature without any contrast agent. And here in this work, basically, Michael tried to analyze what is the advantage of using adaptive optics with OCT and geography. And this slide basically shows you the first kind of large field of view. On the left, on the left, you have AOCT fast images from different depths. On the right, you have just the clinical OCT and geography without adaptive optics. And then on the right panel, you actually see comparison of the same side by side as image with the AO, OCT and geography and just OCT and geography. So clearly, AO gives you a great advantage in resolving the structures, and then it also allows you to create this really nice depth encoded images of, in this case, foveal vascular zone. The next great example of the future directions of AOCT is another work from the Miller's lab. So in this case, again, they acquired multiple volumes as a function of from the same subject over time, and they basically were able to observe that after averaging of multiple volumes, some of the features became visible, and this is mostly due to the fact that in AOCT we're still limited by the speckle. So as we image multiple volumes, and as the structures, kind of ultrastructures underneath even at the structures way below resolution of our systems are changing, they indeed affect the reflections and then affect the speckle distribution, and this indeed in time allows to enhance contrast of some of the structures. And this proved to be extremely useful in imaging of retinal pigment epithelium cells. So here's an example of the data again extracted from the multiple volumes. When you see a single frame, really means a single volume on the left from photoreceptor inner segment, outer segment, and cone outer segment tips, from the rod outer segment tips, and then from the RPE. And after averaging many frames, one can see that clearly the RPE mosaic becomes visible, and one can start resolving the mosaic of cones and then the mosaic of RPE. And after analyzing this data for spatial frequencies, they actually also find out there's a clear peak corresponding to the RPE mosaic. And here, again, you can now create images similar to the ones that were created once with the AO-OCT, except in this case, there's no contrast agents, and basically one can just average multiple. There's also obviously doesn't rely on the fluorescence images, but in this case, purely due to the scattering and then change of the speckle pattern due to the average multiple volumes, so I can actually extract the very nice image of the RPE mosaic and have both photoreceptors and RPE mosaic overlaid and shown like this. And the last from the visual directions I want to show you is it's a very interesting work by Gerhard Huttmann from Germany. They're using somehow interesting technique. This is called full field OCT, and they're actually able to do reconstruct the image of the retina without using hardware adaptive optics. This is they're able to acquire using this full field substrate OCT the whole image of the retina at once, and then by using digital adaptive optics, all computational adaptive optics, they're able to correct for all the aberrations in the system, in the eye, and then basically get diffraction limited images from any depth within the field of view they acquired, within the volume they acquired. And with this system in hand that's extremely fast and very, very stable, they actually were able to observe physiological responses to stimulation from the single photoreceptors. So what you can see on the left is the schematic of the system. It looks really, really simple. It actually is very simple. The secret is a very, very expensive camera that costs probably hundreds of thousands of dollars. And on the right, you see this reconstructed image from the acquired with the system of the volume, AOCT or OCT volume, and then you can see the change in length of photoreceptors outer segment in response to the light stimuli. So light stimuli pattern is showing as this X, and then this is the measured change in length from the photoreceptors. And with this in hand, they basically show that this response is light dependent, and they basically were able to study dynamics. And we also have some of our own work that explain this process as basically the inflow of water due to the change in osmolarity inside photoreceptor outer segment. So I would stop here, and thank you very much for your attention.
Thank you to Robert and Ram for doing a great job of introducing adaptive optics and the imaging. I'm going to switch gears away from OCT now and start talking about AOSLO again. And showing you here the first image on the left is an image of the foveal cone mosaic, and on the right, a more peripheral image showing the rod and cone mosaic. And we saw from Ram that this can be very useful in introducing subjective measures of visual function, as well as trying to understand how the eye is functioning in response to vision stimuli. And Robert talked about uses in OCT to start looking at the photoreceptor mosaic. But there's many other cell classes in the retina, and they've been notoriously difficult to see. But recent advances using non-confocal aperture techniques such as offset aperture, split detection, or multi-offset aperture have allowed us to see things such as the retinal vasculature, the cone inner segments, the ganglion cells here in the bottom left in a mouse eye, or the horizontal cells in the mouse eye. Now, the ganglion cells and the horizontal cells are transparent cells which have been difficult for us to view using traditional reflectance imaging techniques. We've also looked at using fluorescence imaging techniques to look at both autofluorescence and extrinsic fluorophore. So on the left, you see an image of the RPE mosaic. You see the dark nuclei and the bright surround, which show the cytoplasm of the cell. You can also see on the right, though, with the use of extrinsic fluorophores, not only can we tap into looking at cellular structure, but we can look at the function of cells. And here, a genetically coded calcium indicator, GCAM6, has been inserted into the ganglion cells of mice. And in response to visual stimuli, you can see that there's an increase or a decrease in the fluorescence, depending on whether the cell is being stimulated or whether the light's on or the light's off. And this is related to the function of these cells. And so what we're interested in, though, is tapping into the function of both inner retinal cells and outer retinal cells using intrinsic fluorescent markers. And so intrinsic fluorophores such as NADH or FAD involved in metabolism or retinol involved in the visual cycle are difficult to image in the living eye because of the ocular transmission window, which prevents excitation of these fluorophores down in the short wavelengths. However, if we use two-photon excited fluorescence imaging using a long wavelength light out at 730 nanometers, this provides an equivalent excitation at 365 nanometers approximately, which will excite those important fluorophores for metabolism and visual cycle. We can then collect the emission within the wavelength transmission of the eye in the short wavelength visible regime. So to do this, we have adapted an AOSLO to do a two-photon fluorescence imaging. Now, Robin Sharma did this as part of his thesis work. And so this is a typical schematic for an AOSLO. You can see on the bottom left an input source, the Ti-Sapphire laser, which we use for both reflectance imaging and for excitation of our fluorophores. You can see an 840 nanometer source for our wavefront sensing. The light comes into the eye after passing through two scanners, creating the roster pattern and the deformable mirror. For reflectance imaging, the light is coming back out of the eye, passes back through the entire system, and is collected in a PMT shown in the bottom left after having been descanned. You can also see to the right-hand side, there's a Shack-Hartman wavefront sensor for wavefront sensing, which is then being used to control the deformable mirror. Now, differently for two-photon excited fluorescence imaging, we use light coming directly out of the eye. So in green, you can see the light coming out of the eye being reflected directly to a PMT without descanning. And this allows us to collect the light without loss through the optical elements of the system. So we simultaneously collect a reflectance image and the two-photon fluorescence image and use the motion from the reflectance image to adjust for the correction of the motion of the eye and therefore apply those corrections to the fluorescence image. On the left here, you can see a reflectance image, and on the right, you can see the corresponding two-photon excited fluorescence image. You can see in the two-photon excited fluorescence image that there are cones and rods clearly delineated, and that the intensity across these cells is much more uniform than what you would have seen in the reflectance image. You may have also noted this particular cell here, in which it appears normal in the reflectance image or unremarkable, but yet in the two-photon excited fluorescence image, it looks quite dark. The nature of this or the source of this difference in fluorescence is still unknown, but it's under investigation and may be related to changes in the visual cycle of that cell. So I'd now like to talk about 4 different applications of two-photon excited fluorescence ophthalmoscopy. And the first is, again, looking at those transparent cells within the inner retina and looking at the outer nuclear layer, the photoreceptors. So here in the upper left is an image where the nuclei appear dark, and you can see bright surrounds creating the mosaic pattern of the outer nuclear layer. On the upper right is an image in the nerve fiber layer where we can see bright streaks where the Mueller cell processes reside. In the lower left, you can see the walls of the vasculature, likely fluorescence due to elastin and collagen. And in the lower right is the ganglion cell mosaic, almost looking like peas in a pod or strings along a pearl running along the nerve fiber bundles, where you can see the dark nuclei and the bright cytoplasm in the surround. These techniques have allowed us to visualize these cells and allowed us to use, for example, a two-photon excited fluorescence imaging in order to validate images and methodology for the multi-offset aperture technique. We've now then applied this to looking at assessment of the visual cycle. Now, the visual cycle is used for the regeneration of photopigment, and for example shown here for a rod, the photopigment absorbs light in the outer segment, and that photopigment is regenerated through a process that involves the RPE cells as well. Now, the particular molecules involved here are not important to this presentation, but what is the fact that several of these molecules are in fact fluorescent, and that's the two-photon fluorescence that we're tapping into. Particularly in the rod, in the lower left, is the all-trans retinol, and we'd like to look at the production and the clearance of the raw trans retinol in response to visual stimulation. Now, this is important because this process in the visual cycle function can be disrupted in several different diseases. For example, Stargardt's disease, which can produce the production of all-trans retinol. And so Robin, during his thesis work here, identified the photoreceptor mosaic, isolated pixels coming from rods and those coming from cones, and I'm going to show you results for rods in particular, where he tracked the fluorescence intensity over time for all of the rod pixels in response to visual stimulation. So the plot here shows the two-photon excited fluorescence intensity versus time, and we see an increase in fluorescence intensity in response to our stimulating laser for excitation of the two-photon fluorescence. However, this increases and reaches a plateau. At that time, Robin is then able to introduce a brief flash, which will produce, in this particular case, approximately 98% bleach. And that flash introduces an influx of retinol. You can see the production of retinol, followed by the clearance of retinol as the intensity as it reduces. You can see here on the curve to the right. And this retinol clearance, for this particular example, has a time constant of approximately 60 seconds. And so we can quantify as well the magnitude of increase from the equilibrium state before the flash to the initial intensity after the flash as a delta F over F naught. And if you plot that delta F over F naught versus the various intensity combinations used for the brief flash, we can see in red are the normalized delta F over F naught, and in green here are the expected photopigment bleaching levels. And we see good correlation between that. Again, further validating our thoughts that the time-dominating fluorophore in the retina for the photoreceptors is in fact retinol. The third example I'd like to talk about is applying this method of visual cycle assessment to understanding retinal degeneration. And here's Sarah Walters in combination with Bill Merrigan, Botan Roska, and David Williams. We've been looking at using a viral vector injected into the eye subretinally with the intent of damaging the photoreceptor outer segments. Here you can then see in the image on the left the reflectance image. The lower portion of the image shows cones that do not appear to have been affected by the virus. And in the upper portion, we see cones that appear to have had some effect from the virus, in that you can see dark regions with interspersed cones. In the center is a two-photon fluorescence image where you can see the cones clearly delineated in the bottom portion of the image, but in the upper portion of the image we see again dark regions. However, there is still some fluorescence in those areas above the signal-to-noise level. On the far right is the offset aperture image, which is using we can see the inner segments of these cones, and we can see that although the outer segments have been damaged according to the images on the left, here we can see the inner segments of these cones remaining. If we track then using the two-photon fluorescence intensity over time in response to the imaging beam, in blue we see the normal cones, and in yellow you see the cones that have remained within the damaged region. We see the expected increase in plateau in response to the stimulating laser. However, if we track the intensity in the darker regions delineated by the orange area, we can see that there's very little response to stimulation and that this is almost flat, although this intensity level again is above the noise floor. So the imaging that I've shown you can be done with multiple different light levels. We are able to do this at light levels and show functional responses below the ANSI standard. However, if we turn our imaging source up above safe levels and try to look at the earliest signs of light damage, or what are the thresholds for damage, we can start seeing here an example where we selectively damage the S cones in response to the stimulating laser. So in the upper left, you can see 15 minutes following a damaging exposure, the dark cones, and these are consistent with being the S cone mosaic. If we follow this up on moving down, this is an image that's 3 weeks post-exposure at this location. We can start to see that the cones have become very dark and are almost disappearing. If we follow this up several weeks after that, you can see in the image here at 8 weeks, the areas of the circles have started to shrink, and that there starts to be a more random pattern around where the cells used to be. And in the lower right here, although you may have initially thought this was a control location, at 11 weeks we see that the S cones that have been damaged have completely disappeared, and these areas have been filled in by rods, creating an almost normal appearing retina. And so the last example that I want to talk about is just how can we start understanding what the sources of this fluorescence are. And so to start down that pathway, we've been starting to do fluorescence lifetime ophthalmoscopy in combination with adaptive optics. And this is the work of James Feeks and Sarah Walters. Looking at, and the left is the fluorescence intensity image, showing you the rod and cone mosaic. And on the right is the fluorescence lifetime image. This is an indication of the time delay for the fluorescence photons to be emitted from the fluorophores. And we can see here that the cones have a longer lifetime than the rods. And this may be related to differences in the concentrations of the fluorophores, difference in the state of the fluorophores, as fluorescence lifetime is very dependent on the environment of these cells. And we're just now starting to begin to attempt to understand what the sources of these fluorescence lifetime changes are. And so in conclusion, I hope I've been able to show you that two-photon excited fluorescence ophthalmoscopy of using intrinsic fluorophores can provide both structural and functional information that will hopefully help us to understand how the retina is functioning in both normal and diseased eyes. I'd like to thank all the people that have worked on this project in the Advanced Retinal Imaging Alliance in Rochester, as well as our financial support through NIH and through the Research to Prevent Blindness. Thank you.
Excellent. Thank you very much to all of the speakers, Ram, Robert, and Jennifer, for these excellent presentations and for relaying their experience and knowledge to our audience today. So without further ado, let's go on to the live Q&A session. So first question, I think it's probably addressed to Jennifer, is how much power is used for the two-photon eye imaging? Is it safe?
It's a great question. And so in the initial development stages, we tend to use a lot more light than when we're at the stage to being prepared to translate this into human imaging. I showed you one example where we were able to damage the cones, and that was using high light levels over 7 milliwatts for an extended period of time. Typical imaging can be done even below 1 watt within ANSI standards in order to allow us to look at the functional traces within the eye and within the retina of the photoreceptors. So we can vary the light, so it can be done safely without causing long-term damage to the cells.
Perfect. So next question, I suspect it's for Ram, is how can AO help with testing and inventing future vision therapies for retinal diseases?
Thanks, Mark. So I think that on the one hand, we are developing our future vision therapies in a wet lab, on a cellular scale, and studying features of gene therapy, stem cells, optogenetics, etc., on scales which far exceed our current clinical standards. On the other hand, we're testing whether these therapies are working or not on scales which are about 10 or 20 times coarser than the scales at which they're being developed. So my hunch or my prediction would be that adaptive optics would apply to these to testing and developing new future vision therapies, because it would bring down the spatial scale of both testing as well as seeing whether these vision therapies are being applied or working in a human or an animal model, for that matter. So you can take the example for say a gene therapy type approach for say color vision therapy or for LCA, and you can imagine that on the one hand we're developing these therapies, but on the other hand we're not really to the point where we are actually testing the pros and cons of these therapies on a cellular scale. So I think that adaptive optics will really let us accelerate the time that takes from testing these therapies in your lab to when they become available to a patient.
Here's another great question, which is, what are the prospects for translating adaptive optics technology into widespread clinical imaging? Robert, would you like to start to answer that one first, please?
Yes, I'll be happy to start that. So I think that first of all, the AO seems to be very powerful, and really the only limitation right now is the cost of technology and and how easy it is to use it. And I believe that we are, as a field, we're making great progress on both ends. So I definitely see that in the near future, AO-OCT will begin really having a real impact into the clinical day-to-day practice. If any of you guys want to comment that as well.
I think Robert hit the nail on the head there. I agree completely.
And Jennifer?
I really don't have a lot to add. I think the important things for translating some of these newer technologies into clinic are, just similar to the question that was asked of me, ensuring that these things can be done safely and repeatedly in the human eye.
Okay, on to the next question. Which are the requirements for the deformable mirror and for the closed-loop performance for AO-OCT? I suppose that's quite a technical one.
Well, okay, I'll take a stab at it. So understanding the requirements of the deformable mirror first requires us to understand the statistics of aberrations that are present in the normal human population. So knowing that the statistics of the normal human population lets us define the number of degrees of freedom actuators specifically that would be needed to compensate for the aberrations arising from the human eye. And there are a number of papers that have done this, and at the same time, as a wavefront corrector requirement for the deformable mirror, we also need to be able to sample the wavefront using optimal Schock-Hartmann wavefront sensor configuration. So both of those things have to be matched in a way that you are able to measure the aberrations accurately and sensibly, and the DM is able to correct for the aberrations most sensibly. So if you wanted to go home with a number that would be most relevant here, you could think of as a lenslets to actuator ratio of about 2.5 is almost sufficient to measure the aberrations introduced in a human eye and to be able to image the retina at a cellular scale. So those are spatial requirements. And in the early 2000s is an article about the dynamics of the eye's wavefront aberration, how they change, what are the temporal requirements for deformable mirror technology. And it turns out that if you have a closed loop bandwidth of about 4 to 5 hertz, that'd be sufficient to compensate for the changes in aberration that occur due to tear film and other different factors in the eye. And other than that, for a deformable mirror, high linearity is often desired because that makes the closed loop easy and nice to program. High stroke of individual actuators is very desirable so that we don't need to use trial lenses and other spherocylindrical corrections to compensate for the aberrations. Great surface quality is desired because your closed loop performance is only going to be as good as the flattest wavefront that a deformable mirror can make. And finally, hysteresis on individual actuators is also desirable because if you actuate a given actuator and come back to it a certain time instant later, you want for that actuator to behave in a very predictable manner. So that's a short summary. And Robert and Jennifer, if you guys want to add something to it, please go ahead.
No, I think you covered it all. Thank you.
I think that was great.
Okay, so on to the next question. It would appear that all studies have been done in the visible spectrum. Have there been any studies in the near infrared or the SWI or spectrum up to 1.7 microns or higher wavelengths? Thanks in advance for your answer. Maybe
I'll first start and then we can can discuss more. So actually most of the studies with OCT was done in the infrared. So usually it's between 750 and 950 window, 50 nanometer window. There's some work done in the 10, 50 nanometer window. However, if you go kind of beyond that to the even longer wavelengths, the water absorption becomes so high that we actually cannot image retina at all. So we're mostly in OCT, staying in the infrared range, and then for the visible OCT, there's not that many examples. The visible light is usually used for stimulation, but I guess Jennifer can now comment on the rest of the spectra with her techniques.
Well, I think from my perspective, the choice of wavelengths that we're using is highly dependent on what our goals are. So choosing the right wavelengths can have an impact on resolution, but it can also have an impact, for example, in fluorescence imaging on which fluorophores are being excited and what the optimal fluorophores are. And particularly in the visible, we have to be concerned, I think, additionally about light safety, especially at the shorter wavelengths. But a lot of the imaging has been done in the near infrared. A lot of the reflectance imaging is done out at the near infrared. And particularly in the two-photon imaging, we've gone as high as 920 nanometers for excitation, looking out towards lipofuscin and FAD excitation with that wavelength.
Okay, I think Jennifer might have answered this partly in the first question that was asked, but what are the prospects for translating two-photon excited fluorescent ophthalmoscopy into human imaging?
Great question. We are we're really excited about the possibility of doing that. We have recently published a study, Christina Schwartz authored it, on doing the two-photon fluorescence imaging and measuring functional signals at very low light levels that are within the ANSI standard and therefore should be considered safe. We've seen no changes in our animal models in response to those light levels and can do that imaging repeatedly in those eyes. The only effect that we could see, we've measured in response to two-photon imaging, various clinical imaging assessments, and the only thing that we have seen a variation in is the infrared autofluorescence, which is an effect that we know from previous publications that infrared light can have an impact on the emitted infrared autofluorescence, and that happens not only with these pulse lasers, but can happen with CW imaging in the infrared as well. That was a paper by Ben Masella several years ago. And so that currently is our only hurdle, I think, to widespread integration of this into a human imaging system, and we're hoping to be able to do that in the near future.
Excellent. There's one question that maybe I can answer that's popped up, and yes, this webinar will be able to be viewed after the event on an on-demand link, but we're actually well over our hour slot for this evening. You can still submit questions, and they might be answered by email. We can certainly receive them still. But basically, it remains to me to thank everyone that asked questions. Presenters, webinar participants, everybody, thank you very much for your time. Good evening.
Thank you very much.
Thank you.
Thank you.